Okay, welcome to medical students around. On today's lesson, we are going to handle antimicrobial agents. We are going to check on introduction to antimicrobial agents, probably check on these specific agents on the disease mechanism. Now they can be able to act on the disease and maybe accord the act of healing. Now, from the introduction perspective, you just need to understand that antimicrobial agents are widely used drugs, which are starting examples of modern medicine. This is because of a period of time they have been developed to attain remarkable power and all that, the microbial action itself, right? So the entire thing is that there are some probable idea of how we can curb disease mechanism. And so there has been a lot of invention since, but then there are different drugs which are used. Some of them are naturally, some of them are synthetics, and the entire thing is just to help in eradication of microorganisms which are pathogenic or maybe the ones that are unwanted. Now, when you talk about antimicrobial agents, they can be either way. They can be the antibiotics. Of course, antibiotics, they are the ones which are fighting the microbiomes. And you really need to know that most of them are originating from natural sources such as fungi, right, bacteria. So that is what we encode by the word antibiotics. But here we're going to check on the entire thing, antimicrobials, compressive of the antibiotics and any other thing. So the recent development targeted the bacterial and fungal cell was synthesizing enzymes. So the bacterial ribosome, that is included enzymes necessary for the nucleoside synthesis and DNA replication. In a way, the entire thing just tried to test that the drugs, they have to accord some mechanism for them to act or maybe provide that action that is needed. Entirely, you have to know which drug and which organism. And on this organism, how is this drug affecting the organism so that it can be eradicated or maybe killed? At most, we know that the microbiomes or the bacteria or the fungal, they have to have cell wall. They have to have cell membrane. So the entire thing internally, in terms of the cell which are composing the bacteria, also we have some enzymes like which are necessary for various metabolism. So these drugs will just block some pathway like metabolism by substituting some molecules in the metabolites range, or maybe interfere with the cell wall mechanism, so even the cell wall formation mechanism, and by that the organism will end up dying. So the antimicrobial agents, that is at most, so they'll target some part of the microorganism to accord their effect. So microorganisms have the capacity and ability to adapt to environment pressures in many ways, including antibiotic pressures, and this will result in development of resistance, which causes consequences of injudicious use of antibiotics, especially overuse and inappropriate use of the same. So this has led to development of multi-drug resistant pathogens. Entirely, the most important thing is that when we use drugs, when our system, maybe the brothers, when you prescribe a drug and typically or we overuse a certain drug, our body tend to have it in the system, and then the microorganisms which are attacking us will also tend to modify itself in a way that it will evolve to resist the drug. So that is what we call the resistance. So over time, the capacity of the microorganism to defy or to adapt in the environment of those drugs now enable them to acquire resistance. That is what is illuminated by the paragraph. Then we are going to talk about classification of antimicrobials, and we have classification based on the type of organism they are working on, the chemical structure and the mechanism of action, what they are doing. And again, we have independent with the spectrum of activity. So the type also and action and the sources where they come. So we can classify them based on the organism they are acting on, based on the mechanism of action that they are calling, based on the spectrum of activity. In spectrum of activity, we can say either broad or narrow spectrum. Then types of action and their sources from their plants, are they from bacteria, are they from fungi. So classification of antimicrobials, that is what it entails. Now, beginning from the ones, types of antimicrobials. One, we have antibiotics, which I've told you, mostly this term come associated with the origins of the drugs. They come from the other organism, bacteria or fungi. Antibiotics. So again, another one is antiviral agents. They are the ones that attack virus. Now antibiotics normally attacks bacteria mostly, but it is not just bacteria. You can even refer to other organisms as well, as much as the drugs are sourced from the other organisms. So we have antiviral agents which attacks the virus. We have antifungal agents, they really handle fungi. Yeah, we can also call them the antimycotic. Why? Because the mycosis, the mycos, the disease mechanism, the fungal diseases, we call them the mycoses. So can have this one as antimycotic agents. We still have antiparasitic agents. We still have anti-helminthic. Antiparasitic agents, they attack parasites. I'm very sure you know the parasite, which are divided into either the helminthic and the protozoas. So they're all parasite, yeah. If you come to the class of protozoa, the prozoa, right, they are also classified there in terms of their movement. You know all those parasites, the Indolinax nana, the malaria parasites such as the Plasmodium family, all those ones are parasites, right? When you come to helminth, you will know the Vertobutius latum. We have the beefworm. I know you know the Tinea saginata. All of them are parasites. Then antiprotozoal agent, the same, under parasitic drugs, right? So that is that. So we can have antibiotics, antivirals, antifungal, antiparasitic, anti-helminth, antiprotozoal agent as well. Based on the mechanism of action, here we have it inhibits cell wall synthesis, to mean that we have 6 categories of how these drugs attack the microbials or the pathogens. They can attack them through the cell wall synthesis, so they inhibit the transpeptidation, right, deferring some strand from being polymerized, and therefore the entire process is blocked. It can result to what we call leakage of cell membranes, and when that happens, the content which are locked inside the cell membrane comes down. So by that, they also interferes with the entire structure of the microorganism, causing death. They can as well inhibit protein synthesis, and as you know, protein are the chief element that are very, very important in terms of metabolism and even growth of the pathogen. So as much as everything considered, when the drug inhibit the synthesis of protein, even multiplication of this microorganism is going to be rendered, because remember, we have many enzymes which are proteinous in nature. We still have, they can cause misreading of messenger RNA code, and therefore interfering with the entire transcription and replication process of the microbiomes. They can always as well inhibit DNA gyrase, which is one of the most important enzymes responsible for replication of DNA. They can interfere with the DNA function. All these ones will now give us classification of drugs depending with their mechanism of action, how they act. Based on this spectrum of activity, from your hand you can see we have narrow spectrum and broad spectrum. Narrow spectrum are drugs that are specifically targeting a specific disease. For instance, when you talk about narrow spectrum, let's say we can have a typical disease like a drug which are only used for tuberculosis, yeah, and they are not being used for other cases of disease. A specific drug which can only treat malaria but cannot treat any other. But then broad spectrum drug, they have equal effect, right? They have equal effect in a way that they can still treat, malaria, they can treat, the same time they can accord some treatment to the typhoid. So that is what we refer to as broad spectrum. So classification of drugs or the microbial agents in terms of the spectrum of activity depends with all those ones. So either the drug can be a narrow spectrum drug targeting specific or broad in a way. Still proceeding, we can talk about the types of action. We have bacteriostatic and bactericidal. Bacteriostatic and fungostatic. Bactericidal or fungocidal. Bacteriostatic, these are drugs now. We classify drug as bacteriostatic when it inhibits the multiplication of the microorganism or the pathogen. Fungostatic, that is the fungi now is being inhibited. During their division is when this now drug come into play. It can only play when the organism is multiplying. So by that, in bacteriostatic, the bacteria are inhibited from reproducing, so they cannot multiply. Fungostatic the same. Then bactericidal, this one kills the microorganism. Therefore, whether it multiplies, doesn't multiply, whatever, it just causes the suicidal effect. So it destroys the the microorganism. Then let's check on the sources. We can classify again microbials depending on the sources: fungi, bacterial, actinomyces. We can get a source of drug or microbial agent from fungi, like just penicillins. We can source some from bacteria, actinomyces. That is as well, we can get them from those type of bacteria called actinomyces. So let's check on the problem associated with antimicrobials. What are the current problems? Since many years have passed since the evolution and invention of drugs, the question is, what are associated problems with antimicrobial in the present regime? So the problem associated with antimicrobial agents is what I intended before, and I told you that we have resistance, superinfection, and masking of infection. Some antibiotics, and that is what is really a tendency in the external hospital. When you go to the pharmacy, or maybe just say in the clinic, and then somebody prescribes for you a broad spectrum drug. This is a drug which can combat even the symptomatic expression of a typical disease, which is not targeted for or purposed for. So a patient will realize that she or he has recovered, yes, but in this sense, there are some masking of the infection. So it will just suppress the symptoms and signs, and there. So we have a problem to do with masking of infection, resistance in a way that now as a patient, you've consumed drugs several times and you consistently use the drug in a way that the drug now has been so used, the organisms which are portraying your disease, like the bacteria, now are used with the drug, and therefore they formulate a mechanism or adapt with the environment of the drug and overcome the pressure of the drug, and so they survive. Then superinfection is that a drug stabilizes one disease, but again it results to another disease depending on what is adverse reaction, its adverse effect, right? So it can result to a secondary infection as a result of you treating the primary infection. That is what we refer to as the superinfection. Now let's check on the mechanism of action. Every drug has got what we call pharmacokinetic, how it enters into your body from absorption, distribution, metabolism, and even excretion. So on the basis of that, the most key or significant thing you need to know is that the drugs are not the sole instrument of cure, but act together with the natural body defense mechanism, which we call the immunity, to accord the cure. Now, the agents of antimicrobial are different sites in target organisms. So we have the 4 sites which they can target the cytoplasm of the membrane, they can target the protein synthesis, or maybe the nucleic acid metabolism. That is what we had to do with the DNA inhibiting, all that, and DNA gyrase inhibiting as well. So let's start with one mechanism of action. When I say mechanism of action, what is this drug doing to the microorganism? So when you start with the cell wall, for example, in the cell wall, you realize that the cell wall gives the bacterium its characteristic shape and provide some protection, right? So it also helps in multiplication of bacteria normally when they break down and extends. I hope you remember the binary fissions in the high school. So multiplication of bacteria normally involves the breakdown and extension of the wall, and distortion of this process makes the bacteria susceptible. So when the drug acts on the cell wall, so the distribution and multiplication of the bacteria becomes a problem. Entirely it also act and destroy the organism, because it will not be able to multiply. So now that is one circumstances. Now, for instance, we have what we call selective toxicity, which is not here, but I need to know when a drug targets a microbial cell wall, it only does that to the cell wall of microbial, but not human cells. And after all, if a drug targets a cell wall, human cells do not have cell wall. So the drugs that interfere with the cell wall synthesis are effective only against growing cells. And the growing cells, now that will give them the title bacteriostatic drugs and not bactericidal. Example, we have penicillins, they act on the cell wall, cephalosporin, vancomycin, and cycloserine. Those 4 you don't need to forget. Penicillin, those are cell wall inhibitors. Cephalosporin, cell wall inhibitors. Vancomycin, cell wall inhibitors. Cycloserine, cell wall inhibitors. Talk about the cytoplasmic membrane away from the cell wall. You know the component of cytoplasmic membrane. These are membrane inside the cell wall and is the site of microbial cells. So that is that. So drugs that act here interfere with the vital biochemical activities of the microbial organism. That is that. Another key thing that I needed to know is that example of the drug that acts on the cytoplasm membrane, we have the polyenes. Yeah, and in the polyenes we have the nystatin and photericin and the azole family. All these ones are just antifungal. The azole family, we have fluconazole, itraconazole, those are antifungal, and miconazole, antifungal. Then we have another one, the third example, polymyxins. You also need to remember polymyxin because it's a very important drug. Drug which is associated with protein synthesis or protein synthesis inhibitors, we call them, they just interfere with the microbial protein synthesis at various points during the buildup of the peptide chains on the ribosomes. So, like for example, we have chlorophenicol, erythromycin, fusidic acid, tetracycline, aminoglycoside, and linezolid. The other thing is that you need to know this drug with their names. You need to know the mechanism of action and when they are recommended or when they are prescribed as indication. Now, important thing again here is simple, is that protein synthesis, some protein synthesis is very vital, I told you, because even some of the cell organs are just protein. So if you don't synthesize protein in the microorganism, the entire microorganism will collapse. So that is why drugs target protein synthesis, so like chlorophenicol, erythromycin. And you really need to know, apart from the indications or the disease supposed to be treated with, example, chlorophenicol and erythromycin, you also need to know the contraindication and the side effects. The last mechanism of action is nucleic acid synthesis inhibitors. Drugs interfere directly with the microbial DNA or its replacement or repair. Like for example, we have quinolones, the metronidazole, which is commonly known, or this one, they will now interfere with the RNA, like rifampicin for TB, and the drugs may interfere indirectly with the nucleic acid synthesis and then still giving us a same accordant. Like example of such is sulfonamides, the sulfonamides and trimethoprim, the ST. So you need to know that. Now in our next lesson on antibiotics, we are going to do antibiotics when the time comes. Thank you so much, and please do not forget to subscribe in our YouTube channel for rapid and frequent video of such.